Good morning to all the virtual and in-person attendees. It's a huge honor for me to introduce our speaker today, Dr. Miguel A. Mateo. He's going to talk about a powerful ability of coastal vegetative communities to sequester carbon in the sediments. Miguel Angel is a renowned expert in the field of marine aquatic ecology, and he was a pioneer in the research of palaeoecology and blue carbon stocks in seagrass. And he's head of Marine Aquatic Ecology Group in the Spanish National Research Council, and he's also professor at the Edith Cowan University in Australia. And well, I had the pleasure to meet him a few years ago, and I can also, he's an excellent researcher and a marvelous and generous colleague as well. So I would like to thank you, Miguel Angel, for your dedication and support in this Euromarine conference series today.
Muchas gracias. Thank you. Thank you very much, Rocío. I will never get used to this kind of beautiful introduction because I don't feel not even half of what you have said. But anyway, I guess the older you get, the more things you do, and maybe the more experience you have. But apart from this, so for me, it's really very special to be here. It's for me more special than for you to have me, because I feel always with family. I know you since many, many years ago. And coming back to Cádiz University is a pleasure for many reasons. People has to come here to realize all the pleasures around being in Cádiz and in the university. But also because I'm going to present to you a research line that we actually created. We have some problems. I think it's okay, no? I think it's, yeah, to remove some of these lines. I think it, yeah, it's no problem. It was. Oh.
It looks good from our side anyway.
Anyway, I think because the screen is big enough, I think it's not going to be a problem. And also we can see how many participants are there. So we have some extra information. It's very good. 27 is not bad, plus another 10 is very good. No, I was saying that we have the pride of being the ones creating this research line, which is simply realizing that the sediments underlying some seagrasses, actually most of them, these sediments are arranged chronologically very well, so they can make a very good palaeoecological archive. We discovered this, and we have been working and playing with this line since almost 2000, so already for more than 20 years. And it's so much fun. And this is what I want to transmit to you. I'm going to make a little brief account, history of this line, how it was born, and then some of the last or the most interesting achievements or answers to ecological and environmental questions that we have been able to answer and to solve using this tool, which after all is a tool. Yeah, it works. Well, first of all, I was recommended to keep some of these slides where I introduce not only myself, but also the institution. We work for the Spanish Council for Scientific Research. It's the largest institution of research in Spain, and also one of the main ones in Europe. As you can see, is globally in Europe is the seventh, no, global, not in Europe, in the world, the seventh largest research institution. Top 5 in terms of having European Horizon projects. This 11,000 people you can see here refers to all the people that is involved in CSIC, one way or another, which is a lot of people. So it's an institution that is really pushing science a lot in our country. 120 institutes, centers or units or associated units. The institution publishes 14,000 papers a year. We have now about 3,500 ongoing projects. This is our weakest point. We should have been making more patents. I think this is one of the weakest points because it indicates that we are not still succeeding in transference. So what we do in the academia has to be translated into technological advancements, not necessarily, but I would like to have this up. And we direct and we create, say, we give birth to about 600 PhDs every year. Well, now we go quickly. We are in Barcelona, in the north of Barcelona, in the Center for Advanced Studies of Blanes, which is just at the beginning of the Costa Brava, that some of you may know. It's a typical holidays coastline in Spain. Our is about 100 people in total in our center. Is very. This is how it looks like. We're in the middle of a natural park. The ocean is just 5 minutes from there. It's a small place. And then we have these groups: freshwater ecology, benthic ecosystem functioning, molecular benthic ecology, socio-ecological systems in the coastals. This is our group, the group aquatic macrophyte ecology. And we have the recent group is a theoretical and computational ecology, who is a transversal team, who is serving their own interests and, of course, also helping the rest of the teams to model and handle big data series. No? What happened? Because maybe I'm out of the mouse is out of the range. Maybe. No, working. Yeah. So, well, this is the team is very big. I mean, we have been in terms of in time has been very big. Some of the ones are already gone. Some others are not here, but are now working. You can see the senior ones is Paul Ivory, Oscar Serrano, myself, and Montserrat Soler, who is the technician of the group. Many postdocs, sabbaticals, PhDs, postdocs, etc. Some of them are already now in the postdoc positions and so on. This is just a little. There are so many more. No? Anyway, I'm also an adjunct professor at the ECU, Edith Cowan University in Perth, in Australia. That's. I was appointed in 2013, and I'm still one of these professors. And every now and then I lecture also remotely or in person in Australia. Okay, so let's start with the talk proper. Everything started by encountering these structures in the water. No? This belongs to one of the largest Posidonia oceanica reefs in the Balearic Islands. It's in Espujol in Formentera, South Ibiza. I was asked to study the dynamics of the treated compartment in seagrasses, and then we started to dive around and take small samples and so. And then we took advantage of these walls, no? So we could sample here, 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 and there, and then do some analysis. And then is when we began to realize that this was a highly organic structure with a lot of organic carbon and organic matter and carbonates. We wanted to date these structures and realize that they can be, these ones were like 2,000 years old, but then in time we realized that we arrived nowadays even 9,000 years old of history recorded in these structures, with the property that it was very well arranged. So you could nicely see that one strata was overlying the older one. So this was matching one of the main conditions that a nice paleo record has to accomplish. Nice. So the oldest down, the youngest up. And this is a nice life lesson as well. No, the youngest has to go up, and we have to just begin to relax. Let's say, not get buried, but relax. Anyway, so taking a close look to the structure in the upper sections, you can see a very entire material. This is almost 90% organic matter, roots, pieces of rhizome of Posidonia, and then 3,000 years later, in the basis, you can see still a lot of Posidonia fibers and even almost an entire rhizome here, 3,000 years in age. So this immediately told us that the preservation conditions in this structure was very good. And this is when we began to think about using this as a paleo record. Why is it so important? Because in the coastal zone, in exposed areas, we don't have any other paleo record. You have to go offshore, very deep in the ocean, to get a very nice record or sedimentary record that is being deposited nicely without perturbation, disturbance. But in the coastline, it's not possible because it's very dynamic, unless a species like this comes, roots, fixes the sediment, and keeps this chronological structure. And then we saw this huge opportunity to have a nice, precise record with a nice resolution. So we published this seminal paper. This is considered the first paper in blue carbon and in paleoecology. With colleagues from the Smithsonian Institution as well, my PhD director, Marta Perez, another colleague. We made the first numbers, accretion rates, carbon, nitrogen, and phosphorus accreted gram of sediment per square meter and year, and so on. We made the first modeling. This is the cumulative mass. That's the age until 2,500 years. And this is the model. And as you can see, in the end, you will have an asymptote. There's no more accumulation. And this is the maximum accumulation capacity of organic carbon of these reefs that you have seen. So we gave first numbers, no? So you can have up to 4,000 tons of CO2 equivalent accumulated per hectare in this Posidonia meadows. Of course, we had no idea that this was going to be called organic blue carbon. This happened in 2009, no? So imagine this 1997. And also we modeled that, and this is really amazing because it matches perfectly with reality. The residence time could be at most 12,000 years of age, which is the time all the material takes to decompose, but also the time when the sea level rise stopped rising, and therefore Posidonia kept on growing, and then there is no literally possibility of having mats, these reefs, older than this age. So, in other words, this is more or less the temporal window we can aim at by using the palaeoecological approach with Posidonia, with seagrasses, about 10,000 years, which is the Holocene. It's a very important period because it's when things are happening. Well, things are happening always, no? So it's a long-lived reservoir, makes a real sink, an ideal sink, and has information from the Holocene period. Do other seagrasses form these mats? Yes, but remember that was 2 meters. This is 3 centimeters. This halodule. Well, they make little mats. Underneath this mini mat, you have, of course, organic matter, but because the production of the plant is not so important, and also because the material is not so recalcitrant, they do not store for so long time. Okay? That's a detail of this mini mat of halodule luminaris. And this is Posidonia, and this is the reason why you have this. Once the leaf detaches in autumn, all these leaf bases, the sheaths, they remain, and they are extremely recalcitrant. So together with the roots, this is a default input of organic matter in the sediment, which immediately consumes the oxygen. The leftover labile materials creates immediately a very anoxic environment, and then boom, it serves the whole structure. And that's why it takes thousands of years to decompose. This is an amazing view. This can be more than 1 meter rhizomes that you could really do a lepidochronology with them. It's amazing. This is the reef, and this is one of the impressive images. Imagine all this is called now blue carbon, is the picture they use to impress people. Blue carbon, look, it's massive, it's fantastic. And this is very quickly how these structures grow. They begin to elevate the sea bottom. This is the rock, and it keeps on growing until it reaches a steady state, where the same amount that decomposes, or until the sea level rise, so they cannot course, live outside of the water. So this is the limit of the thickness. And then we can see more or less the age, the stratigraphy, the thickness, and you just go and take a core, different sections. This is what we wanted to do. We wanted to not to be limited to sample reefs that we could see. We just wanted to go in the middle of a meadow, core it, take a nice virgin piece of sediment to do this palaeoecological approach. This is a fetish bay for us, Port Lligat. Is where Dali, the painter, has a house here. So we thought, let's do something surrealistic. And then is when we, so all this is Posidonia, all these patches. Posidonia goes from 5 centimeters depth until 25, going out of the bay. So it's a nice, healthy meadow. This was the famous Core 2000, taken in 2000. This is the view of the bay. That's the Core 2000 taken. And this core yielded almost 15 publications with just one core because it was so novel. How did we sample this? Scaffolding from, you know, these structures they use for painting the facade. They build these metal structures. No? So we use this in 2 small rubber boats. We just fix them with truck clamps. A commercially drilling light platform or a tower. And then we use steel barrel with a PVC barrel inside. This is the core catcher, the core catcher. It closes when you pull the core up. And a Roadworks self-powered hammer. You know this. Okay. So with this, we managed to take 5 meters long core. This one. This is the famous Core 2000, section 1, 2, 3, and 4. So 4 maneuvers. And this is how it looks like. You can see the very organic layers, and then almost, no, I was going to say almost mineral, no. Even in the basis of the core, there was a lot of organic matter from Posidonia. Okay. There you are. For me, it's very emotional to show these things because it's a long time ago and it was so exciting, no? So we wrote this seminal paper. The International Atomic Energy Agency invited us to present that. You may think, why? What does Posidonia have to do with International Atomic Energy Agency? It's because it was about stable isotopes, and we used already some stable isotopes analysis to get some information in this long-term archive. Right? So that's why they wanted to. So what we wanted to do in this paper is to present in society the new paleo record. And we made sure to state that these deposits are a very good and loyal chronological sequence. The species is abundant in the Mediterranean, so we can do geographically large-scale studies and comparative studies. The time span was the Holocene, which is very good. The resolution was very good. You will see later on. They have so many things in there. If you take a slice of this core and then you open it on a tray, you can see so many things. And I can study all these things, how they have changed in time. And of course, many things that you can't see, like chemical analysis and ratios of many things. No? And also, it was the only possible paleo records to be used in exposed areas where waves change all the structure of the sediments and make it impossible to use these sediments as a paleo record, except for the places where Posidonia is growing. So what I wanted to, the authors were Javier Romero, a PhD student of mine, and a colleague experienced in isotopes, etc. No? I saw this in a commercial in an airport. That was a stock market company. Our long-term view improves our view of the short-term. So of course they claim that because they have experience and they know how does the market react when things happen in the world, like Ukraine, Russia war, etc. They have experience and they know how the markets are going to react, so they can advise you, don't buy now because it's going to go down, wait until. So this is totally, you can translate this into the ecosystems. If you know, have experience on how do the ecosystem change as a response of different disturbances, how long do it take to recover, which is the trajectory they take when this perturbation happens, then you can, of course, advance and recommend not to leave the ecosystem go into this direction, because this can happen, because we know, because we have seen in the past what happened. So this long-term view is extremely important to make wise decisions when it comes to manage our environment. This represents simply an ecosystem. It's just one ecosystem. It's a typical old school way to represent an ecosystem. It's not many, it's just one. The ball is any variable that you can imagine, is, I don't know, temperature, productivity, anything. They have a natural mean value, is the attraction point, but they can, of course, vary depending on season or whatever, little perturbations and so on. But they always tend to have this position until, for some reason, a strong perturbation, they can go and surpass a threshold and then change state. Changes in state are common, can happen, have been happening naturally, but also now we are forcing to make many changes in the ecosystems. Mar Menor. Mar Menor was a crystal clear water, benthic dominated, and now is a muddy, turbid water with a water column domination, dominated productivity. Plankton versus benthos. But this was a change of state. The shape of the cuvettes are the resilience. The deeper they are, the more difficult it is to change them, the state, etc. And these are very like precarious states that can change with just a little bit of perturbation. So one ecosystem can have different states. And we will see in the last example of paleo reconstruction, you will see a beautiful example of an ecosystem that has changed over the last 6,000 years. Time arrow, how can we study this? Well, so the more you open the observational window, the time scale, the more you know about the possible states the ecosystem can reach and where are the critical points. Okay. You can monitor this, but it's costly, it's not so resilient. You cannot maybe monitor many variables because you know how expensive is this, but you can also complement monitoring with paleo reconstruction. So the information has already been written in different languages, and you can translate these languages and learn about the ecosystem. And the goal of this approach, palaeoecological approach, is to have more predictive capacity for the future. Now, ways of coring the meadows. Piston coring, this is in the Seas Islands. This is only good for very soft sediments. Here is a small video where you can see different ways of coring. Vibrocoring, maybe some of the top centimeters of the vibrocore will be a bit disturbed because of the vibration. This is a drilling platform that can be submerged. It didn't work at all, this one. For some reason, it cannot really keep the material inside. You will see now how efficient is the vibrocoring. Of course, you immediately can realize that the logistics are pretty bon. So it was 20,000 euros a day to have the boat, the crew, the geotechnical people. Yeah, I mean, this is like a dream. You can do this once in life. And then you retrieve the core, and its advantage is 6 meters in 30 seconds. No effort. That's how it looks like when you have a core, manual core. This is manual coring here. You see a PVC pipe. I want to see the last bit of the video. Yeah, just manual banking. Pam, 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 underwater. And there you have 1.5 meters or 5 meters. Of course, you will see it's a video. Sophia's laughing because she's seen it. There. So you can also bank 5 meters course, which is maybe 6,000 years of archive. But you need strong people. You need to go to the gym, or you need to be young and strong. So you can afford this kind of something. Of course, you have to cut in sections the course for shipping purposes. Now, this is once we have the core, we have to know the age of it. We combine lead-210 and carbon-14C, AMS, accelerated mass spectrometry with just 1 gram of sample. You can date, or even less, you can date have the age. You combine for more recent times, the lead-210 sometimes is more efficient. And then for the longer periods, more than 100 years old, you can use the 14C. I wanted to share these graphics because this is all in the same meadow, Santa Maria in Cabrera, at 5, 15, 10, and 25 meters depth. This is the age, 200 years, 600 years, 4,000 years. So in the same meadow, depending on the depth, if you focus here, 80 centimeters core. At 80 centimeters, you have 50 years in age. At 80 centimeters, you have 1,000 years in age. So even in the same meadow, the growth rate, the elevation rate of the map, of the paleo record, is very different. So if you are aiming at recent changes and you want a lot of very good resolution, you should aim at fast-growing mats. If you want to reconstruct much longer periods of time, you want to go to very slow-growing mats to capture a much longer period of time. Okay. So that's what I wanted to show you. You have to know about your. So this is the range we have found. Mediana and Y. This can be from 0.5 to 4.5 millimeters per year of growth, with resolution of between 2 and 20 years per centimeter per slice. You can have only 2 years of this fast-growing meadows. So you have to use one record or another depending on the address. I'm going very slow. We have the archive. We know how old it is, but now we have to learn what it's telling us. And for this, we use what we call the proxies. The proxies can be anything that you can imagine. Proxies are limitless. You can use new ones, create new ones, think of new ones. They can be, of course, the age, just the aspect of the lithology. You can use optical and UV and any kind of imagery that you can imagine, magnetic susceptibility, X-ray fluorescence, basic descriptors of the sediment, of density, granulometry, sedimentology, organic matter, carbonates, nutrient contents, how many sheets I have, how many rhizome biomass, if I have found or no peduncles. Well, this applies to lepiochronology. Yeah? I'm not going to talk about this. Stabilizers, pollen, pigments, non-palynomorphic palynomorphs, pigments, animals that stay there, forams, diatoms. All this is preserved in the sediment, even the DNA and the pigments, photosynthetic pigments, they stay there. This is paleothermometer. This is to a paleothermometer, so we can know the temperature changes along this period. Glomalin is a land use change indicator that we developed in Australia and then we used in here, etc. But this is just a little So how does it work? I'm interested in some environmental parameters, how they interact with the biological parameters. This is typical, no? I experiment, I observe. And I also realize that some features, they co-vary and they kind of represent this change. For instance, stable isotopes, we know that are an indicator of productivity in the plant, primary production, stable isotopes of carbon. And we know that productivity is a function of light. So light, productivity, delta 13C or delta 15N is the proxy. If we analyze and we establish this relationship between this trio, these three elements, and then we take this proxy, analyze it in an archive, here we produce transfer equations, real time, present time, use the transfer equation, apply to the proxy retrieved in the paleo records. So I analyze pieces of Posidonia 3,000 years old. So I can reconstruct a relationship of how was the irradiance or the productivity 3,000 years ago. That's the basis of palaeoecology. So we published the first seminal paper with the first paleo reconstruction using Posidonia oceanica, showing that the meadow was very stable. This was the different light treatments we established, growth, productivity, light measurements. And we realized that in time, the productivity was a function of light reduction. You can see with all this also, we realized that there was a nice relationship depending on the light treatment. The delta 30C also reacted very well. With this, we created transfer functions. And with these equations and the paleo delta 13C, we managed to reconstruct irradiance and net leaf production along 1,000 years, showing that it's quite stable. No big changes, no big trends or shifts. So this is good news. Second example, and I have some examples. When I run out of time, I just thank you very much for your attention, and we go for a coffee. Anyway, so this is a PCA performed with all these heavy metals. We analyze the heavy metals along the core because most of the metals, they have the same behavior. It was nice to make a synthesis of them by making a PCA. So we got this factor 1, the one in red, which represents those metals that are typically human-derived pollutants. That normally in nature, if human is not participating, they are in a lower amount. So we realized that we found like 3 phases, or if we own 4 phases or 5. 1 phase, 2nd phase, 3rd phase, another phase, and then the last one that you cannot see very well is like a much higher increase of this synthetic metal pollution component, F1. So one of the important things in palaeoecology, or the answers that they provided is, have you ever been asked by someone, hey, imagine that tomorrow we wake up and we have no idea about anything in biology, nobody knows anything, and goes to La Caleta. Wow, we are biologists because we remember that. You know, we remember. Let's make a nice bionomical study of the La Caleta, which species are there, and you will find Rugulopteryx, no? Wow, so this is species from here and so on. You have no idea that it's an invasive. You will say, this is normal. It's the normal composition of. So have you ever been asked by any manager, hey, these levels of pollutants or this productivity of the plant, is that normal? It's okay, it's wrong, they have to be worried about this? How can you answer that? I was asked by the director of the national park with the Thies Islands, Miguel, because I got this natural parks project. I want you to answer in this project, which is the natural vegetation of the Thies Islands. Because we did pollen analysis in the Sidonia record. So we can tell which was the, and I told you, look, Pepin is the name. What do you mean normal, Pepin? I don't know. Yeah, the normal composition of, you know, the vegetation has been changing always. So you have to tell me, when do you want me, I mean, which frame of the movie you want me to tell you about? I will give you all these landscapes you saw in this drawing of how the ecosystems change state. This is the whole ecosystem. What do you want to be here or in this longer cuvette or what? Oh, well. Wow. So in the end, he said, well, before the humans began to mess up, which is a nice reference time. So before we begin to transform our, so we are with this palaeoecological approach, we're in the condition to say what is not normal, but a reference state, the one we want to come back to. If you don't have this frame, this long vision, you cannot tell. So we saw these two peaks. One was the Greek Empire, the pollution of metals, their industry, the Roman Empire, and the Anthropocene slowly increasing, and then the Industrial Revolution just peaking a lot in them. So as you can see, this is a powerful archive. It's recording with loyalty what is doing the humans. What are the humans doing with the environment? And this was a nice finding, because here we can say that 2,800 years ago, humans began to be noticed in terms of metal pollution in the Mediterranean. Until then, no impact, boom. And then non-stop until now. That's another interesting information. When did we start to pollute the Mediterranean? 2,800 years ago. Anyway, we published this in Science and Total Environment. If you want to ask, you can ask. I just put in the mic. I like very much this example that I was astonished with this. Core 2000, 4 gigawatt arsenicum, a very dangerous pollutant. That's the pattern of changes. No? So I realized that contrary to any other pollutant, arsenicum began to decrease. That's strange. The other ones were just, peaking like crazy. What happened with this? No? So we dated it very well. This Industrial Revolution, of course, the pollutant increased, but then in this date, it began to decrease significantly. We went to the pollen paleo record and realized that exactly in these points, when it begins to decrease, the Vitis vinifera pollen grape disappeared totally. And it's exactly when the phylloxera destroyed all the grape yards in Europe. You will say, what does have to do with the... So people was fumigating regularly the fields of grapes with a pesticide that in the past was basically arsenicum to kill the animals, but not the grapes. No? So of course there were no grapes because the phylloxera, the bug ate everything away. There was nothing, so they stopped fumigating. And that's why using historical records to date the outbreak of the phylloxera, a pollen record and a biogeochemical record, the metals, arsenicum, we could reconstruct the history of what happened and why the environment is now being cleaned from this pollutant. I think it's very pretty. Now, this will be the second last one example, and then this example, another one, and then we are done. So hold on. Australia. I, of course, exported this with Oscar Serrano, exported this palaeoecological approach and research line to Australia. And then the South Australia Public Water Company, with authority in monitoring the quality of the waters, told us, look, we know about this approach you are using. We want you to go to this place, Port Pirie, in South Australia. It's one of the counties. We have Queensland, South Australia, South New Wales is one of the south counties in Australia, because this is a huge problem. The kids are having brain problems. They are being totally unsuccessful at school. They noticed that a long time ago. So the problem is that the levels of blood of lead in the blood of the people are extremely high. Okay? So this was the reason behind, and this is because the Belgian company Nistar has the largest lead smelter in the world in this county in Australia, an enormous, and it's emitting lead in the atmosphere like since many years ago. So they told us, look, we want that you give us proofs that, because they claim, no, it's not our fault. This is a naturally rich area in lead. That's why we are here, because it's naturally rich. They say, no, it's not us who are polluting, no? So they say, can you use your palaeoecological approach to demonstrate that this began to happen when they started? Of course. So we went to Australia, went to Port Pirie and took course. This is published. Ana la frata serrano pera masque and Paul Library. So I don't have to explain this slide, no? Time, calendar years, VP, just to 3,000 years ago. This is already calendar years. This is when they built the smelter. And this is the lead concentration, normal, reference, boom. These are the two cores we took. And these are changing points, significant changing inflection points. So I call this forensic paleoecology. I can tell you why did the system of the individual died, because so they were very happy. So we say, well, let's take another course far away from Port Pirie, because maybe the trend is the same. So this Moreton Bay and North Harbour is so much, 2,000 kilometers away, at least from Port Pirie. Normal, well, a little bit, but normal. Port Pirie. So no discussion. Yeah, but no. So we made stable isotopes of lead. The black dots are the course we took in Port Pirie. This is the signature of the two ratios of the lead. The yellow and the blue is the raw material they use in the smelter, the raw material to extract the lead. So it's totally attached here. If you go down in the course before the smelter was built, then you have another signatures. Before 3000, so it was absolutely clear that they were the ones producing the lead that was in the atmosphere was coming from their smelter, because it has the same signature fingerprint as the raw material they use. So there was no discussion, and now they have this proof, but politically they cannot do anything. It's something that they tell us, but they cannot say that publicly. They prefer the people to die slowly or to have problems rather than die because they cannot work. So what do? They analyze the lead in the blood regularly, and when you reach a level, they send you out of the country for some months. When you are purified, you can come back. That's the solution at the moment. Money talks, but they have the proof. Now nobody can deny that. But the governments, none of the governments, no matter which is the color of the governments in power in Australia, they never dare to close that smelter. Microplastics. Almeria, a lot of plastics. Before the plastics, they realized that the plastics make the crops much more efficient, imagine. When the plastics were, this is a Posidonia core in Cabrera, in Almeria. So in the late 70s, they began to pollute, and the Posidonia record began to record the presence of. And now finally, this is the Stiers Islands. We took a core. Remember this platform I showed you at the beginning. Now this has collapsed, so there's no connection between the open ocean, very small connection. So the tide takes hours to change here after it has already changed outside. So very, very low turn over, the waters get so overheated. This is a sandbar. So this is a closed lagoon, almost closed lagoon. This is being declared the most pretty beach in the world some years ago. It's very pretty. So in summer 2000, they make this picture of Zostera marina in the lagoon. Everything covered by Zostera marina, and 23 years ago. It took course, 6,000 years, organic matter, no organic matter, no carbonates, peak of carbonates declining, density of the material, grain size. Well, I'm a bit late, so I'm finishing. Stable isotopes to tell us the origin of the organic matter, carbon content, nitrogen content, the core picture. You see very deprived of organic carbon period, an enormous amount of organic carbon in that period. This is between 2,000 and 6,000 years ago. So you can see how with different proxies we are recreating what happens. This is the history of this lagoon. Manganese redox potential indicator, proxy, organic matter proxies, pigments, which kind of plants were dominating there. Water column, freshwater plants, terrestrial plants, seagrasses. So with this, we did the reconstruction with beautiful pictures. 6,000 to 4,000 years ago. 6,000 to 4,000. That was a freshwater coastal lagoon. This is the open ocean. The open ocean to the west, and this is to Vigo, to the continent, Spanish continent. The dune I saw you, and then there was start to be some organic matter deposition coming from the watershed of the island, and this is the aspect. Could have been the aspect. This is a recreation. 4,000 until 3,500 years ago, so coming to the present. Thick deposits. That black period I saw you in the fall is very black and organic period. Pit, freshwater pit. The dune was kept on growing. Sea level was rising, dangerously, not dangerously. And then sea level rose, inundated, flooded the lagoon, turned into a marine environment, coastal lagoon. Zostera colonized because the island is surrounded by Zostera marina, meadows, beautiful ones, but they entered and colonized the lagoon. But then, because they built this passerelle and the passerelle collapsed and the water became almost stagnant, temperature rise, nutrients for the camping began to rise, the ecosystem totally died. And this is what you see at the moment, just nothing. A few fish that can come through it's collapsed the connections that they still remain, but then it's basically some algae, and that's it. So you have seen changes from freshwater lagoon to peatlands to mixed sandy dune systems with some freshwater ponds, then into a nice seagrass dominated ecosystem, and now just a very almost azoic environment because of a very heavy environmental impact. That was the last example, 50 minutes. If you want to play with this research line, be ready to work with many different experts all over the world who can help you to translate the languages in which the proxies are written, because all of them are a world in themselves. And it's so much fun to contact different colleagues and they will collaborate for free, no? Just for the fun of making these reconstructions, no? And yeah, so that's all I wanted to explain to you today about this special line for me. And I only remain to thank you very much for being here, and also the ones that are in remote. And if you have any curiosity, any question, I'm absolutely happy and ready to answer. Maybe they are too tired of such a long time.
This interesting contribution. For virtual person, if you have any questions, you can text in the chat and we can read them. And here in person, there's some questions. Okay, here. Okay. Gloria.
Thank you very much for your talk. I really enjoyed a lot. I have a few questions related with that, because you explained that you can make a reconstruction of the environment depending, for example, how much was the light. And as you said at the end, you need a lot of multidisciplinary people to interpret this record. And I was wondering if you has been working with experts on sediment dynamics, sea level records, or things like that, to be able to not only depending on the sediment record, but also on the modeling of what happened before.
Well, we've done, for instance, to know all these changes, the sea level rise, we were with oceanographers that knew about this sea level changes. And so we've been working with geotechnical people who help us with the coring. For instance, the palaeothermometer, the temperatures we have reconstructed was by a microbiologist in the NEOS, Stefan Schouten, who created this proxy 5 years ago. So he realized that the lipids of the membrane of the archaea, they were more or less saturated depending on the temperature. So, wow, this can make a proxy, paleoproxy. So he developed it, and we used it, and we have been able to reconstruct the temperature in the Balearic Islands since many years ago until now, and you can see how it's basically getting colder, the water, but now it's already goes increasing. What else? Gabriele Procaccini, he helped us with the eDNA, to study whether the plant was reproducing vegetatively or sometimes sexually. So by using stuff he uses, this kind of RFLPs, and so he can tell us whether this is a new clone or this is the same clone. And if it's a different clone, it means that there's been a sexual reproduction. And so, what else can I tell you that can help me? Of course, we're in the need of taxonomists, because the wealth of organisms that are kept there is so huge. And we have made some master thesis where we found invasive species. So you have the nice record of foraminiferans and ostracods and so on. And all of a sudden, there's one that only appears a few decades ago. Boom. And then we checked in the data banks of invasive species, and yeah, it's an invasive species from the Red Sea, a little snail in the Balearic Islands. And so you can see when it started, and you can see the population dynamics, how it increases and how it stabilizes. So all these different experts that you cannot really take everything. It's impossible. So the fun thing is to make large groups, slice your course, and this slice, sub-slice it, and send it out to the different. We send the slice frozen to Canada for the pigment analysis. So and geologists, of course. Yeah, and people knowing how to also use echo sounding to be able to tell us in advance if this Posidonia meadow has a nice mat underneath. So we'll have to try, they're hitting the rock, so they can, with bottom profiling, they can tell you the thickness of the mat, which helps also to evaluate the carbon story. So it's an absolutely multidisciplinary discipline. You can, of course, just analyze your little foraminiferans. That's it. It's very important, very useful. But no, it's not so much efficient.
Thank you. And I have a second question. It's about being able to trace from where comes the organic matter that is in the sediment. is enough with the stable isotopes, or the signature of stable isotopes change once it arrives to the sediment?
This is a very, very interesting question because, of course, I didn't extend it in the explanation, but the proxies are good proxies and bad proxies. A good proxy is a conservative proxy that we call them. That once it is laid in the sediment, it doesn't change at all, ever. For instance, more or less granulometry. If mud arrives, mud stays. Organic matter. Is that a proxy? When it arrives, it begins to slowly decay. But the decay process is quite predictable. It's an exponential decay. So you can detrend your profile, and then what stays is the environmental signal. Stable isotopes, we have all the data. The paper is almost written. I'm waiting for some student to go for it. And we have studied the conservancy of the different proxies, metals and stable isotopes. The delta 13C in 300 days of decay remains constant, 300 days. Talk about 3,000 years. We don't know. It is accepted that it's conservative, stable isotope signature. How can we know? So this is a very good question. We need to know how robust our proxies are when it comes to memory reconstruction. So, but the good thing is that the multi-proxy approach is used because the different proxies will have overlapping information. So there are more other proxies that also tell you about the production or the origin of the organic matter. So one is confirming the other, and then you begin to be confident about your conclusion. The multi-proxy approach is very important. The uniformitarianism principle is that we accept that all the processes that are happening nowadays can be totally translated 10,000 years ago. And maybe the Rubisco carboxylation enzymes of the plant were working differently, and they were processing differently and discriminating the stable isotopes differently in 10,000 years ago, maybe. So we are reconstructing the past with tools that are not perfectly the same as it were before. So this is the uniformitarianism principle. That this means that we accept that these processes have not changed much in the time. We cannot assure it. So.
Okay, thank you.
Do you?
Well, Rebecca is texted in the chat. Where will the meta pollution have come from during the Greek and Roman era, and what kind of metals are they?
Thank you for the question, Rebecca. Actually, it was in the image. There was so I don't know, iron, aluminium, chromium. No, this is more environmental. Maybe we can retrieve the. So yeah, it's very well characterized which metals are typically from the metal industry. So one of the, you know, that the Romans and the Greeks, they were very strong in metallurgic activities. You can see zinc, lead, cadmium, copper, iron. These are not elements that are easily removable naturally from the. I mean, they are of course removed, no. And the ones in blue will be the more, say, environmentally natural elements that you can find in the nature. So, and this is very well established, more or less, no? So that's why the Romans, they had this very, very strong metal industry. And also it's very interesting to see the palinograms, how the impact of the warship building had an enormous impact. They had to almost clear cut all the forests everywhere. In Spain, the Pyrenees were almost totally bare. So they just had to collect wood to make the ships, no? Mm-hmm. So you can tell, because many species disappear or decline a lot, the pollen of these species. And also the glomalin tells you that decreases a lot, telling you that it came from the land changes from forestry to naked land or cultivated lands, no? So So yeah, they had a real important impact, and this is the dark ages, the middle ages, where things were a bit like, okay, now we are not so much doing things, no? But then slowly everything began to start again until now that we are now out of range. So it's basically mining activities, and these elements you can see there, plus others that we did not analyze, because these were analyzed not with fluorescence, but directly digesting the material, going to the ICP-AS, and analyzing the exact content of the quantity. So this is expensive. Anyway, so yeah, these are the main methods we use for this construction.